maybe before we talk about this new topic. So, any quick questions? Okay, if none, so we end up discussing how you generate BCR and TCR diversity, right? So which involves having multiple germline genes, VDJ recombination, junctional flexibility, P and N, uh, region nucleotide addition, and eventually the combinatorial diversity provided by light and heavy chain to form the paratope, right? Obviously, for T cells, so you don't have this process that we call as somatic hypermutation, or right, which you actually find in BCR. This was the last slide we talked about. The next thing actually is the antibody engineering. Okay, so why does one have to engineer antibody? So that is first question, right? Anyone? And when we actually say antibody engineering, what do you mean by that? Am I audible?
Yes, sir.
Yeah, but then why you guys are mute? So no one is able to say anything, even remotely. Yeah, say it.
So antibody engineering is actually the process where we are actually artificially like developing the antibodies having a particular specificity. So if you want to target a particular disease, so actually if we are trying to increase the pool of antibody particularly, so in that case we engineer the antibodies. So.
Yeah, that's one of the most important point. Because conventionally you generate these antibodies by immunization, right? Or and with the immunization, what you do actually is so you have induced clonal expansion, right, of your cells. And when you do so, whatever the secreted product of these antibodies you get, so you really do not know out of these trillion molecules or billion molecules, so which one actually is specific to what antigen you are, because this actually is a bulk preparation, right? So and you have multiple specificities available in that. In order to get an antibody against a particular antigen or particular epitope, yes, you can resort to what we already covered, which is monoclonal antibody generation, right? Where you have a clone of B cell, and then you fuse it with a cancer cell. Once you get this hybrid, so the antibody production actually is coming from this B cell, right? And immortalization is coming from cancer cell. And then you use those conditions where you actually can select only the hybrid cells, and then screen what does it produce, and then you get a basically cell which can serve as almost inexhaustible source of this antibody supply, right? So through monoclonal antibody technology, you can actually hybridoma technology rather, that's the right name. So you can generate antibodies of a particular specificity. Okay. Antibody engineering usually is when you modify, okay, part of your antibody or at genetic level. Okay, so this actually does not happen at a protein level once you already have produced this antibody. So this is at genetic level. And why do you have to do it? Because sometime you have to increase, uh, as Vishnu pointed out, you have to increase the, for example, production, the yield. That's one. Second is sometimes you have to make it more stable, right? So that it can resist those biophysical and biochemical uh, conditions which actually are not favorable for proteins to work, right? Sometime you have to actually engineer it in such a way that you can make use of this antibody for one purpose or the other. For example, if you want to make it as a diagnostic antibody, so you would want to label it with some dye, that's one, or with some enzyme, that's second, or with some agent uh, such as toxins. Okay. And why do you actually require that? For example, if you're targeting some specific cells, or if we talk about in clinical utility, if you are willing to target cancer antigens, so you generate this antibody against those antigens. Okay, but if you actually modify it to put some other structure to this antibody like toxins, so it will go and bind to that antigen and it will try to kill it by different mechanisms, right? The other thing could be that you actually want to achieve neutralization of these pathogens, and maybe the antibody that you started out with does have the specificity but does not bind to the antigen with more uh, affinity, so it has a less affinity. If you want to make it more affine, so then you actually have to tweak with the constitution of this antibody at genetic level. You have to modify some of the amino acids where basically you know that these are actually involved in protein-protein interaction, right? So for all these processes, actually you have to engineer your antibody uh, to endow it with the function that were not usually possible if you don't achieve this by engineering, right? As such, when we talk about antibody engineering, we are talking about genetic engineering. Okay, and essentially what that means is basically the locus that you're targeting here, or the sequence that you're targeting here, is for the antibody molecule, right? And this is the purpose why you have to generate this engineered antibody. So the other name actually for this process is called veneered. Okay, rather the center we have gotten here at ICER for antibody engineering, actually its name is veneering technologies. Okay, veneer means basically modification, right? The other thing is sometime you have to actually reduce its immunogenicity. See, initially when these antibodies were actually made, the monoclonal antibodies were made, they were generated in mouse, right? And maybe you have demonstrated that the mouse antibody is able to bind to a human antigen, maybe present on some cancer cells, right? And you can inject it to basically clear this in this developing cancer. But somehow, because of injection of antibody from a different species in human, from mouse to human, so this is going to actually generate some immunological reactions, right? And you know what epitopes would be recognized in this case, because you're talking about two different species, right? Right. So you have all those determinants in the antibody antibody molecule, you have allotypic determinant, isotypic determinant, idiotypic determinants. All those things can serve as antigenic epitope against which human can start generating antibody response or T cell response, right? And if you do so, chances are that in first injection, nothing is going to happen. But if you do it for the second time, and invariably when you actually have to perform a cancer immunotherapy, so you have to actually give multiple shots of these antibodies, multiple injections of these antibodies, either at the site or systemically, right? Um, After first injection, so if you generated antibody against this antibody that you actually injected in animals or in human actually in this case, but the source of this antibody was animals, mouse, right? um, Obviously those antibodies that you have generated are going to bind to this antibody. They generate immune complexes, and then you start getting all kind of hypersensitivity reactions, anaphylaxis. Okay. um, Obviously, one of the conventional approach is serum therapy. Basically, when you inject this antigen of choice in horses, collect serum from horses and inject in humans, as is done for treatment of intoxication or envenoming. Okay, snake bites, dog bites, or whatever. So, in order to avoid those reactions, you have to modify this antibody in such a way that it actually has minimum immunogenicity in the host species in which you are injecting it. Okay, that is one of the actually very critical aspects of antibody engineering. Okay, apart from its modification to make it better suited to one function or the other. Okay, so that is why you have to actually do antibody engineering. For example, so you start with a mouse antibody, and you know that the most of the epitopes in antibody molecule were in the constant region, right? So somehow, if you keep the same antigen specificity, okay, which was actually encoded by VDG and VJ recombination in mouse, but you change this constant portion. Am I still audible?
Yes, sir.
Okay, someone actually tried to log in. Um, but anyways, so you change the constant region of this antibody in both heavy and light chain, right? Your antigen binding portion remains from the heterologous species, but you know that idiotypic determinants are, if you actually look at the immunogenicity, the maximum immunogenicity is present in the constant region. So whatever is present here, so these are more or less idiotypic determinants. Okay. Very low immunogenicity actually is associated with the uh, variable region of antibody molecule constituted by both heavy and light chains, right? So rather than injecting this antibody for therapy, so if you inject this will be better tolerated, right? Because this will have lesser level of immunogenicity in the injected species. The other way of achieving or making it even better is because you know that the areas, the portion, the regions of antibody which actually interact with your antigen are these three CDRs, right? That we call as hypervariable region. That actually determine the antigen specificity of an antibody molecule. So you keep those CDRs as same. Okay, so basically those will be for human origin, mouse origin, the CDRs. But everything else you can make it from human origin. Okay, so in this case, this would be called as a chimeric mouse human antibody, where actually you have one portion of uh, mouse and a large majority of it is coming from humans, which will have less immunogenicity. So in this case, okay, just a second. Hello? Yeah, Hello. Yeah. Yes, I did. Okay, so yeah, so in this case, basically what you do is you keep the region which interact with the antigen same from a heterologous species, and you change everything else. Okay, this approach actually is also called as CDR grafting. Okay, basically where you actually get the CDRs from uh, an antibody or the sequence of antibody which is known to have that kind of specificity, and you basically graft it on a human framework, right? So basically everything in this antibody is of human origin except for the CDRs, which are actually coming from a different species. Now this actually will be uh, almost uh, unimmunogenic in the injected species, in this case human, right? So these are one of the uh, these are actually few of those things that one has to do during antibody engineering, so to make it less immunogenic. The background actually we already have talked about it. That monoclonal antibodies, they are actually used for many clinical reasons, anti-tumor antibody, for instance, right? And if they are actually developed in a different species, so they are going to cause uh, reactions. Okay, if it is of mouse origin and injected in human, so this is called HAMA reaction, human anti-mouse antibody reaction, HAMA, HAMA reaction, right? And in order to minimize it, so you actually have to humanize your antibody. This is called humanization. So you can either make it chimeric or you can make it a completely humanized antibody, right? Um, how do you go about actually doing this? So that was one of those things. But the first thing actually has to be, first you have to have the sequence of this antibody, which has a particular antigen specificity, right? This is the normal approach that you use. For example, how to make this chimeric antibody, right? Or monoclonal human whatever the antigen antibody. Okay. Uh, for this also, you can actually use a mouse, but eventually the antibody that will be produced will be of human origin. And how this is achieved is through this process. So basically what you do is so you take basically a mouse, right? And you hormone prime this mouse by injecting this um, uh, PMSG and many more hormones, so that you actually get uh, hyperovulation. So and from there you can actually derive embryonic stem cells, right? So you isolate ovaries from this hormone primed mouse, um, and then you can generate embryonic stem cells. Now, in these embryonic stem cells, you have to inject um, the the genetic region, the loci of heavy and light chain. Now, where do you inject? You have to inject it in these embryonic stem cell which are already knocked out of immunoglobulin gene, right? Because the aim of this whole exercise is to generate antibody in a mouse which is of human origin. So that means this heavy and light chain genetic locus has to come from human, but eventually, so you are making this antibody in mouse, right? Um, so there is a way of knockdown of this immunoglobulin gene from these embryonic stem cells. There are different approaches. You have CRISPR-Cas, you have shRNA-mediated knockdown, and many more approaches. Okay. Now, these loci, which have a particular specificity for heavy and light chain, so they are actually put into this human artificial chromosome. So this is actually a vector, but it can carry very long sequences. Normally, if you use a plasmid, so you cannot really clone long sequences, right? And you know, the whole locus for heavy and light chain in a rearranged B cell actually is quite long. It's quite lengthy. So you basically inject the whole locus. It has intron, exon, everything, right? Um, and basically, then you put this human artificial chromosome, which has these sequences of heavy and light chain into these embryonic stem cells, right? There is a process to do that. Once you have generated these stem cells, embryonic stem cells, which actually now are carrying um, genetic locus for heavy and light chain, right? So they are actually now put into this blastocyst. Okay, so you can actually isolate. This is called blastocyst injection. Okay, you have these embryonic stem cells, and the next step in this whole process actually has to be what? You make this blastula, gastrula, morula, so all those stages, right? So actually you capture them in blastocyst stage, and in this blastocyst through micromanipulation, so you inject these embryonic stem cells which were carrying sequence for human heavy and light chain, right? Now, if you inject this blastocyst in animals, okay, or whatever the animals that you are going to generate from this blastocyst, okay, so you are going to first generate chimeric mouse. This actually is mouse, but um, it will have some sequences of human origin, right? And these sequences actually are coming from heavy and light chain gene loci of your antibody molecule, right? So you generate chimeric mouse. Then this chimeric mouse actually is interbred on a particular background, right? And when you do so, rather you also tend to actually cross it with a particular HLA background, okay? HLA transgenic mouse, so that you don't have the compatibility problem, right? Eventually, what you are actually trying to do through this process is that this mouse, now after this interbreeding, is able to produce antibody against that one particular antigen, right? Against which you actually started out selecting this heavy and light chain. Okay, so you have this mouse. If you inject with this antigen, so almost all these spleen cells that you get from here are able to produce antibody molecules. Spleen means B cells. Okay, and for example, if you have bred it on a rag deficient background, so it will have only B cells, right? Because you already have incorporated immunoglobulin gene. So it will have immunoglobulin in these B cells. And then the other processes of rearrangement are not going to take place because they are on a rag deficient background, right? So you collect spleen cells, you fuse them with these myeloma cells, and these myeloma cells are going to produce these antibodies. Obviously, if you actually inject this antigen, So this mouse on a reg deficient background is going to make a lot of these antibodies which are specific to the antigen one. Okay, but mouse has a lifespan. So after whatever 2 years or so, it's going to die. Okay, unless you actually keep on breeding these animals on that particular background. So uh, it's okay. So you can actually collect these antibodies from serum, and that will be highly enriched in antibodies against that particular antigen. But eventually you follow the same process as was done for um, generation of these hybridomas, right? You fuse these splenocytes with myeloma cells, and whatever the hybridoma you select, so they are going to produce antibodies against this antigen X. This is just to make this subsequent step only in the laboratory condition. So you don't require animals at that stage, right? So this is how you generate um, human monoclonal antibodies against an antigen 1 using this mouse. Now, you can ask that why can't we actually collect some some PBMCs and fuse it with these cancer cells and then try to select those specific antibody producing cells? You can do that, and people actually have done it, right? But the cells that you get from PBMCs are very limited, particularly those cells of a particular antigen specificity. And then it's a very tedious process. But if you follow this, so your chances are very high to get these different clones which are going to recognize this antigen with one affinity or the other affinity, right? So that is why you actually have to practice this. So is this process clear? All right. Okay, so there is another approach that actually you can use for generation of this antibody. Okay, uh, essentially what that involves is you start with a population of B cells or PBMCs, and then you actually have to select a particular clone, or uh, this actually involves generation of libraries in the first step. Okay. Uh, Libraries of what? Libraries of the variable region of heavy chain and variable region of light chain. And now you know that in your antibody molecule, so you have these two of this variable region of heavy chain and two of this variable region of light chain. So that means, so this these four domains that you are going to use. One is VH, two of those are there, and VL, two of those are there. Because on one side you have one arm, another side you have another arm. So what you actually try to do is you try to make single chain fragment variables. Okay, so basically all these variable region actually are present in a single chain. Okay, so how do you go about it? Yes, obviously you start with these B cells. Okay, and because you actually have to amplify these sequences, so even if you start with PBMCs of that species, it will work, because your primers are only and only going to amplify immunoglobulin gene. Okay, and you have to choose it in such a way that it only amplifies the variable region, right? And why? Because you already know that in this variable region you have the framework and then CDR, then framework 2 and CDR, and framework 3 and CDR, and then framework 4. So essentially you have to design your primers from the framework region 1 and framework region 4, both for light and heavy chain. Now there is little complication here, particularly for the light chain, because you have these two, kappa and lambda. So if you want to generate a library, so you have to do it for both kappa and lambda, right? Um, Essentially, if you are able to amplify that product, right, you can put it in any vector you want. Usually what you do is, so you actually put it in a phase mid vector, right? And once basically these sequences are put in phase mid vector in fusion with one of the coat protein of this M13 bacteriophage, which is actually commonly used bacteriophage, right? So when this virus actually is produced, so P3, the surface, this is one of the coat proteins. For example, you have this bacteriophage, and coat protein is basically on the surface of this virus particle, right? Now, these antibodies, because you have used this phase mid, where you used the sequence of your antibody and a part of, or basically the sequence for one of these coat proteins. And this is actually coming out as a fusion product. Now, the antibodies will be displayed on surface of these bacteriophages, right? Now, if you have an antibody which is displayed on surface of a bacteriophage, so it's easy to actually screen what it is binding to, what it is not binding to. And once you know that this sequence is specific to your antigen of choice, you can elute, you can actually disrupt this interaction with that antigen, right? You still have this bacteriophage, right? If you infect your bacterial cells with this bacteriophage, so this is going to amplify that sequence. And in so doing, so you actually can select an antibody recombinantly using this phase display approach. Okay. Obviously, what you do actually is you isolate gene segments for V8 and VL domain from your B cells. Let's say uh, these are heavy chain and these are light chain. Okay. And then basically, if you want to make it as a single chain, so you actually have to put some linkers, right? You have to actually put a linker sequence here so that this can be linked with the uh, heavy chain variable region, right? Uh, and then you let basically this linking process take place. And apparently you have to understand, because this linker will not decide that this variable region of the light chain has to combine with this variable region of the heavy chain. So this whole process is going to happen in a very random fashion. So initially you might have less number of antigen specificity, but because of this random event of combining or linkage, so you can actually increase the diversity of your antibody. This also has a potential problem, right? We can talk about it, but the process remains like this, right? So you have the sequence for variable region of the heavy chain, and you also have the sequence for the variable region of the light chain, and they are linked using some either natural or unnatural linker, right? And once you do that, so if you basically dimerize in the sense, then basically you have the another segment. So now you have basically in one construct have produced all the variable region of the antibody molecule. This is called single chain fragment variable. Obviously, you have to put these linker sequences, and these linker sequences have their own problem. They have their own advantages, disadvantages, but you have actually brought variable region of the heavy chain and light chain together, right? Once heavy chain and light chain variable region are together, and if you are able to refold this molecule well, so actually you have the antigen binding fragment here. The antigen, because SCFE is going to bind to your antigen because you have created this paratope. And if you modify it in any other way, so you can try doing that using all these approaches available in the antibody engineering. So you want to make it bispecific, bivalent. You want to make it even multivalent. So you want to make it as a dye-tagged antibody and an enzyme-linked antibody, or maybe a toxin-linked antibody, right? This is basically the process. So you actually have generated this gene library for SCFE constructs. Okay, you come up with this bacteriophages, right? And what I said was that you basically use this phase mid in which you put the sequence as a fusion product with the coat protein of one of these virus, this bacteriophage, right? And when you do so, and you try to actually transfect, or rather this is called transform, right? Transform your bacterial cells with this construct, you are going to produce these virus-like particles, or rather bacteriophages, right? And on surface of these bacteriophages, we are going to display these SCFVs. And once they are present on the surface, um, obviously they are present in bacterial cells, so you have to do many steps to recover those specific antigen binder. This process of selecting specific binders which are displayed on surface of this bacteriophage against your antigen of choice is called biopanning. Okay, essentially what this means is, so you do first round of biopanning, so you get the specific interactors, right? You elute this bacteriophage, you multiply it by infecting bacterial cells, then you again perform this biopanning process, but at this time you actually reduce the concentration of your antigen which was actually coated on the surface of this plate, and you do this process for 3 or 4 times, so that uh, because with every single round, so you have reduced the concentration of your antigen. So that means only the high affinity binders are going to interact. Low affinity binders are not being selected in subsequent steps, and eventually you can generate a very high affinity antibody displayed on surface of these bacteriophages. But you already know the sequence for this VH and VL actually region are present in the bacteriophage, right? The genome of the bacteriophage. You amplify that sequence, you put it in a bacterial expression system or a yeast expression system or a plant expression system, and you try to produce this antibody in whatever the system you are interested in.
Excuse me, sir.
Yes.
Sir, in the previous slide, when we were showing the SCFE structure, you talked about the linker peptide. So, sir, whenever we talk about any linker, we associate it with the covalent bonding that is happening. So, are
Well,
we?
that actually is covalent. So the linker sequence actually present in at the genetic level. Okay, so you have a sequence. So basically, it can have this leucine sequence. There are different types of. You can also have it from the tubulin protein, right? So you can also get it from the hinge region of the antibody molecules. That also actually is little flexible, right? So there are different ways to get this linker sequence. Essentially, all these things are happening at genetic level, at nucleic acid level. So you have variable region, then you put a linker sequence, and then basically you have the variable region of the light chain. This whole thing now actually is present in one construct that you actually start out here. The next thing that remains actually is these antibodies are displayed on surface of these virus particles. Yes, Yudish, you have a question.
Yes, sir. So
First,
here,
just a second. Anuradha, is it clear? Or Aradhana?
Yes, sir. Aradhana, thank you.
Yeah. Yudish.
sir, in this flowchart in the enrichment step, so here we are incubating on our desired antigen and basically removing the population of phages which have like non-useful antibodies. That's what is being done here.
Yeah, exactly. So one thing is because you are starting with a library which has maybe million or billions of these clones, right? And your antigen will interact with only select few of those. You start with a library, but from this library, you actually have to select one particular book. And that book basically, in that sense, means the bacteriophage which display antibody for the antigen of your choice. Right?
Sir,
So this,
and in the
yeah.
final step, you where it is labeled phage of desired antigen specificity, can
Yeah.
we be sure that this particular phage has like 100% covered with our desired antibody, or does it also have like mixed with other combinations?
Well, because you have amplified only a single bacteriophage, right? Through multiple steps. So you already know that this bacteriophage, because you are starting with a single bacteriophage and you are amplifying it by infecting bacterial cells. So whatever the new virus particles are going to be formed in bacterial cells, all of them are going to display this antibody of your antigen specificity. Right? There are subtle issues with this approach, fine details, like you have to use ambassador pressure strain of a bacterium, so that actually it can read through this thing, and when you actually put the sequence in a bacterial expression system, so you only produce this antibody molecule. So all those are fine details, but steps are basically enlisted here. Yes, the short
Yes,
answer
sir.
to your question is yes.
Okay, sir. Thank you.
Okay, and now if you are able to express an antibody in a bacterium or a yeast or in other systems that you are interested in, so you can make quintal of it, right? And if, for example, it is specific to a very relevant antigen which can promote immune responses, which can kill cancer cells, you can make billions, literally. Yeah, well, obviously it has to go through many steps. That it's not toxic, it's not immunogenic, and it's able to achieve that desired outcome. It can control that cancer, and these cancerous tissues can be efficiently controlled, and for how long and for how many shots you have to give. Obviously, for all these things, first you have to do some animal experiment, and then you go to clinical stage. And if you get a antibody which is relevant for one cancer or the other, so you are a billionaire. No?
Sir,
Yeah.
I have a question which might not be related. So, can cancer cells develop resistance to antibodies? And if yes, are they more like easy to develop resistance to antibodies than drugs?
Well, your answer to your question actually is yes. Okay. And this we know from the normal carcinogenesis, right? So you don't develop cancer just like that. First, your immune response induced against those cancerous antigens is able to control it to some extent. So this is called basically the elimination phase. So you are developing a cancer because of mutation in your cells, which are undergoing division and so on and so forth. You have as these cells have altered their antigens, so you induce immune response of cytotoxic T cells, B cells, and all those things, and they are able to control it. So this is called elimination. Second actually is equilibrium, where cancer is evolving and your immune system is also evolving. So you whatever the antigen you already have, so it's able to control those, but then you can actually get some escape mutants from these cancer cells, and then they try to cover it up. They try to make big cancers, and then your immune response is again going to induce antibodies or cytotoxic T cell response against those new antigens, right? And in so doing so, you actually achieve a stage of equilibrium. Third stage is basically escape. So where your immune system is exhausted, it's not able to work out to clear this cancer, and then you succumb to it. Okay, so there are different stages. And how this is happening, because cancer tissues, when these cells are dividing and generating these cancer tissues, so they keep on also modifying their antigens. So selection of antigen actually is very, very critical. You actually have to choose an antigen which is not normally present on your cells, present on cancer cells, and it remains more or less constant during the progression. And if you are able to hit it hard in the very beginning, chances of your survival are much higher. So that is why actually many of these cancer immunotherapies, they can prolong your survival, some by 2 years, some by 5 years, some by even 10 years, and in very rare cases by 10 or 15 years, right? But eventually, unless you come up with a combination therapy, so it becomes difficult. So that is why the field of cancer biology or cancer immunology actually is it's being difficult to deal with. A lot of people are still dying of cancers. Yes.
Okay.
Yeah, Vishnu.
So in contrast to the conventional therapy, I mean, therapy where we use both the both the constant and variable region, here we are solely using the variable region. So
Yeah.
due to this case, is there any drawback to this approach?
Well,
Because
yeah, well, I understand your question. So for example, if you use a monoclonal antibody, so obviously if it is from the same species, it's good. But then the level you can produce this antibody in, right, using your hybridoma, cell factory, cell culture approach. So that is always limited. And when you keep on using this antibody, obviously you're not having any flexibility to modify this antibody to tackle these mutants which are arising. That's another one, right? Once you have only the variable region sequence, then actually you can modify it in a way that we talked about. You can actually put a toxin so that this antibody come and bind to this cancer antigen, and then this toxin actually is able to kill this cancer. So all those things are not possible if you use only the monoclonal antibodies, and they are not engineered in any way, shape or form. You have to actually rely on your endogenous mechanism like ADCC approach, which can be, yes, very well done with the constant region of the antibody, right? Complement activation. So you have to rely on those mechanisms, phagocytosis. But if you have to use an approach where you are utilizing this portion to bind to the antigen, to either, for example, if it's microbial in origin, so it can utilize it, it really does not let this thing to infect your cells. And for cancer cells, so it goes and bind to it, and because you already have put up something else, that is why these are called actually as magic bullets, right? So you actually, but bullet, if it is not targeted to that specific organ or tissue, so you are going to get problems. But with the use of antibodies, you can actually do a very efficient targeting.
Fine, sir.
Okay, so this table actually enlists many of these antibodies which actually are used for treatment of different cancers or infectious diseases. Well, predominantly these are actually cancerous antigen, but in this case, palivizumab, this is against this RSV infection and it's used in children. Okay, some of these examples, and let me tell you, each one of those antibody actually has more than $1 billion market, each one of those. It's not like a single isolated antibody. So if you are able to make one antibody which bind to an antigen and it is relevant, it's causing something, it's able to improve survival either during an infection or during carcinogenesis, it's a big deal. Okay, so the next thing that we are going to talk about is antigen-antibody interaction. And these are the objectives. So we have only 10 minutes left. Rather, it's already 50. To describe the nature of antigen-antibody interaction, okay, to compare and contrast antibody affinity and avidity. So these are two different terms. What is the meaning of affinity? We have used it quite frequently when we talk about the antigen-antibody interaction, right? How does it differ from avidity? And basically how to delineate whether this actually is a specific interaction or specific reaction, and this is a cross reaction. And obviously then we are also going to use some commonly used tests for which involve antibody molecule, right? So this is what we are going to talk about in this basically topic. Now tell me, so do we continue with this thing, or because this is a new topic, we start next week?
Sir, maybe we could start it next week.
We can start it next week, but let's say for next 2 minutes or so, if you have any more clarification from whatever we covered in this week, so very quickly we can go through those things.
Sir?
Yeah.
I didn't quite get why the chimeric mouse must be intercrossed with a HLA background mice.
Yeah. So okay, just a second. So interbreeding means that, see, this mouse is a wild type mouse here. Okay, so you have a wild type mouse. You did a lot of this hormone priming and stuff, and once it was basically able to have this implantation in the uterus, so you inject this blastocyst, right? And blastocysts are the one which actually carry the sequence for your antibody molecule, right? Because of this transfection that you have achieved. So once this mouse, right, is able to have this implanted blastocyst, right? So in this case, so you have a heterologous sequence from a different species, in this case human, but as this is a mouse, right? So that is why you call it as a chimeric mouse. Now, if you breed it on a particular background, right? So some of these animals are going to lose out that sequence, because it really depends on, because your this sequence actually is inserted into the chromosomal locus, right? And these are diploid. If it is basically inserted in one of the locus, so the other chromosome, the corresponding uh, the diploid set, is not having this. And when you breed it on a particular background, so some of the animals that you will get from this breeding will not have any sequence. Others will have it, right? And then you actually have to screen which animals are having this sequence and which are not having this sequence for the antibody molecule, right? And that you can easily do it just by taking a piece of tail and then amplify using your PCR specific primers and so on and so forth. The other problem that could happen with this mouse is, because this is a wild type mouse, it has all these rags and other molecules which are required for rearrangement of the endogenous loci, right? If you do not interbreed these animals on a rag deficient background, so the cells that you might end up getting will produce this antibody of human origin, but also of mouse origin. And then the whole process actually is a waste. You cannot really get these B cells, which are making only a large amount, rather most of these clones, which are actually making antibodies of uh, basically derived from the sequence of heavy and light chain that you started out with. Okay, so that is why this interbreeding is very, very important. And then you also have to ensure that you have a germline transmission. Let's say if you have only this thing inserted in chromosome and it is not basically passing it to sperm and ova. So eventually, so this thing has to stop with this mouse only. It has to pass to the next generation as well. You have to make sure that you have to have a germline transmission. And if you look at what is the frequency of having a germline transmission, this actually is very low, sometimes actually even less than 1%. So you actually have to do a lot of screening for this process. Well, that is why I thought this process actually is much more simpler, because wherever you are working, so it's all in a laboratory condition with some vector which you can select against antibiotic and all those kind of things, right? But it has its inherent problem. If you can address those problems, so this actually is a better way of generating antibodies than the conventional approach where you generate these monoclonal antibodies by hybridoma technology, or with this embryonic stem cell or blastocyst injections. You get the idea. So why do we have to interbreed? Because we have to one, ensure germline transmission. Second, that you do not have rearrangement from the endogenous locus, because then it will confuse. So maybe some clone will make it for human origin, some will make it for mouse origin, right? And this is going to basically destroy the whole process. it will make your life so difficult that you cannot really select these antibodies in a more convincing manner. Okay, if no more questions, so we are going to actually end this here. And on Monday, we are going to have a quiz only on the part that we covered after your mid-sem, first mid-sem. And then obviously, we are going to have your questions, and then we'll try to talk about this antigen-antibody interaction. And in the last 50 minutes or so, we'll have a quiz. And I'm pretty sure you must have received your grades.
Yes, sir.
Yeah, okay, all right, fine. After this first mid-sem, until today, what we have covered, so that will be syllabus. Syllabus for second mid-sem, I will discuss that when it comes.